Hey everybody, Mr. Murphy here. Let's wrap up Chapter 4 with Section 4.5, which is all about extracellular structures. Right off the bat, extracellular means outside of the cell, so keep that in mind. And in eukaryotes, we have two major components of extracellular stuff, and that is prominent fibrous macromolecules and a gel-like medium with different fibers embedded. So when we think about cells, we are made out of trillions of cells, but those cells aren't simply the only thing that a human is made out of. There's plenty of stuff in between those cells, enabling those cells to have different functions and also supporting those cells so that it's not simply every cell is exposed to some sort of external environment that could be harsh or damaging. In plants, the cell wall itself is an extracellular structure, which may be surprising because we don't ever really talk about that, but it should also make sense. It's outside of the plant's cell membrane. And we know that it's largely cellulose-based, which is the fibrous component. But then there's also this gel-like matrix that contains all sorts of cross-linking proteins and things that hold it all together. And here's a diagram of that. So we're looking at a plant cell, and here's what's called the middle lamella. That is just this stuff that's sort of there in between the different components of the cell wall, and this would be like the cell wall of one plant cell and the cell wall of another and a third cell wall here, and so it's linking these things together and sort of filling in the gaps. So if you think of like these different cells as being like different tiles on a floor, the extracellular material is forming sort of the grout that's holding it all together and filling the space, so you don't just have emptiness between them. This has major roles. Plant cell wall, I think we primarily understand it's providing support. Cells in a plant undergo a lot of stress and strain from the weight of the plant itself, and so they can help that cell to maintain that shape despite those pressures. It also acts as a barrier to infection, so the cell wall actually has lots of holes in it. You could kind of think of it as a bit like a Swiss cheese. However, it still does serve to protect the cell itself from larger harmful materials and potentially dangerous pathogens and things. And then it also contributes to the general form that cells or plants grow into by sort of defining this shape that the plant will grow to fill. Different plant cells are connected through channels that are called plasmodesmata. We'll see how animal cells do very similar things in different types of cells, but this just allows movement of materials from cell to cell so that there's a plants don't have the same level of vasculature like arteries and veins that animals do, and so this is a mechanism by which the individual cells can be sharing the essential things that they all sort of share a need of between each other. And so even things like RNA and proteins can sometimes sneak between different plant cells. So it really is a connected network that is working together for the good of the larger organism. So animal cells, the outside of our cell, we consider that the ECM, or extracellular matrix. So that's an important term to just be aware of, and we'll use it vaguely often just to sort of refer to the stuff on the outside. The fibrous component is the protein collagen. You may have heard of collagen as like something that people sometimes get injected into them as like smooth out wrinkles or whatever, because what happens as we age is that collagen begins to deteriorate and we decline in the amount of collagen that we have between our cells, which is why your skin starts to sag or whatever, and you end up with wrinkles or other lines associated with that. The gel-like matrix has proteoglycans. That's not a term that I'm ever going to ask you about, so it's useful information for sure, but not particularly to us. But here you can see a kidney cell and a blood vessel, and within that sort of barrier where these things meet each other are this network of extracellular materials. And so that's basically there as support. It helps to link these things and hold these things next to each other because, for instance, with a kidney cell, there's lots of filtrations that's happening between the blood vessel and the kidney cell, both directions. And so we need very close association between these things. You can't just have your blood vessels sort of loosely nearby and hope that your ions in different materials could actually jump across the gap of nothingness that would be between those cells. And so there's a pretty clear function to this network that's holding things together. Here's some more pictures of the specific like collagen fibers and things, but again, not specific knowledge that we're going to be quizzed on or anything. So its role here, it holds tissues together. We'll talk about the different ways that it does that. Helps to contribute to the physical properties of cartilage, skin, and other tissues. These are supportive tissues that take on very specific shapes for their own functions. And so the extracellular matrix sort of helps, kind of like the cell wall, to define what the shape of these things are. It can help to filter materials, orient cell movement during growth and repair. And so there's a protein called integrin that actually helps with the movement or just linking in general. And here's an example of this. So inside the cell, here's some actin, a microfilament, part of the cytoskeleton, and that's linked then to this transmembrane protein that's going across the membrane and then has this integrin that grabs onto the extracellular matrix. So if this is a type of cell that sometimes may need to let go and move or shift its orientation in some way, then what you would see is that integrin would experience a shape change, which is perhaps caused by ATP, bonding a phosphate to it, or something has changed about the structure of this protein in order to basically unlink it from the extracellular matrix. The last part of this section goes into these ideas of ways in which cells join one another, and we call these cell junctions. There's 3 types that we look at in this class, and this is information that you should be aware of. So the 3 types are tight junctions, desmosomes, and gap junctions. In reality, there's multiple types of tight junctions and multiple types of desmosomes. It's just endless examples continuously providing exceptions to whatever nice consolidated list we want to create. But these are the 3 sort of big classes of how cells join together that we should be aware of. Tight junctions prevent substances from moving between cells. A great example would be like in your bladder, right? Or your stomach. These are organs which hold different types of liquid or substances inside of them, and you really don't want those liquids just sort of seeping between the cells. You think about like the bladder, which holds urine. There's a lot of water, different ions. There's urea, which is a nitrogen waste product. But basically, those are tiny, tiny molecules. And so when we have 2 cells next to each other, we don't want to just rely on these cells like, stay next to each other, hope they do, because water and ions and stuff like that is just going to fly right through the gaps that would inevitably exist between those. And so tight junctions are a mechanism by which those cells are basically firmly attached together. Desmosomes, meanwhile, hold cells together, but they do allow some materials to move in the matrix between them. And so this is an example of like a little bit more give to the interaction between those different cells. And then gap junctions are a very specific example, which we'll see in heart cells in particular, where they're similar to the plasma desmata, actually allowing molecules to flow directly from one cell to another. So they still hold those cells together, but it's a channel for materials to pass through. And here's a great diagram which shows you all of these things. And so here we have 2 cells which are stitched together with tight junctions. That would not allow anything to pass between these 2 cells here. And it would be just a whole network of these intermediate filaments that are tightly winding these 2 cells together. So proteins basically stitching the 2 cells together. Desmosomes are associated with cytoskeleton components as well. There's some cross-threading across these 2 cells, but it's got a little bit of space for things to sneak between. It's not as tight as the tight junction, and then gap junctions here, which are like these little pores straight from one cell to the other. Tight junctions a little bit more closely here. You can see this network of stitching so that if something's coming from up above here, it's going to bump into basically a line of these proteins, which are going to pinch the 2 cells together closely enough that those materials hopefully won't seep through. here is the desmosome a little bit more closely, and you can see it's not a big long stitching, but rather these little singular points where the cells are held together. So stuff could still seep around this between the two cell layers, but it's still holding these things fairly close together. And the last one, the gap junctions. Again, linking two cells together, so it's holding them in place, but these proteins are spanning actually both cell membranes. And so they have a hollow opening, or wouldn't necessarily be hollow, but it would be a particular shape to allow certain types of molecules to pass between, but maybe not necessarily everything, or at least it may differentiate based on size to allow for ions and things to pass. Like in the case of the heart, the coordinated beating of the heart means that different ions associated with the muscle contractions need to be able to pass from cell to cell to cell, which is a really cool example. The heart's not specific to the curriculum of AP biology, but that coordinated effort is the reason why we get contraction first in the ventricles, then in the atria, then in the ventricles, then in the atria, and that delay is mediated by gap junctions. At the beginning of this chapter, it presented a question which was asking, what do modern cells tell us about the origin or early cells that existed on the planet? And just to return to that, the things that we've been discussing, particularly the aspects of the phospholipid bilayer, have been explored through this concept of protocells. And protocells are synthetic human-made cells that demonstrate the simplicity of the chemical requirements to create the environment that is the cells we see today. And so basically, it's chemically possible to create the conditions of phospholipids existing in an artificial environment and allowing them to interact just based on their chemistry and arrange themselves into membranes and even into bilayer membranes, just because of the polar and nonpolar interactions, and how we've talked about it in class, but the importance of that establishing of an inside that is different than the outside, and how that affords now this chance for change, which is what biology is really all about, is chemical change happening that's driven towards some sort of functional purpose. And so, if something like an RNA molecule was inside of this protocell, and this protocell has formed a bilayer around it protecting that RNA, now this RNA has a different sort of chemical environment in which it can potentially replicate itself, begin to manipulate or alter other molecules within that cell. And this is really the foundation for some big ideas that we'll get into when we get to our evolution unit, all about first life theories and the potential origin of genetic material being RNA, not DNA. So it's really cool stuff, and we will return to it. All right, that is chapter 4. As always, let me know if you have questions.